Imagine searching for a single Lego brick in a massive bin. This is what scientists face when isolating one protein from thousands in a cell. Proteins are the tiny machines that keep us alive, but to study one, you have to separate it from a cellular soup. Protein purification is a huge challenge in biotechnology. Your target protein might be less than 1% of the mix, like finding a needle in a haystack made of needles. If the protein isn't pure, experiments fail and medical discoveries slow down. For years, purification was slow and inefficient, with much of the protein lost along the way. This bottleneck held back research and new treatments. Scientists needed a tool that could act like a selective magnet, gentle, adaptable, and precise. The solution came from an unexpected place: seaweed. From seaweed, scientists discovered agarose, a remarkable substance. Agarose became the foundation for a new era in protein purification. It offered the selectivity and gentleness researchers needed. And that's where our story takes off. Agarose is a purified sugar from red seaweed, often seen in science labs or even desserts. When mixed with hot water and cooled, agarose forms a 3D mesh, a microscopic sponge. Scientists shape agarose into tiny porous beads, each a maze of tunnels. These beads have a huge surface area, perfect for catching proteins. They're chemically stable and gentle, keeping proteins intact and functional. The watery environment inside mimics a cell, protecting delicate proteins. By adjusting agarose concentration, scientists control pore size for different proteins. Bead size can also be customized for specific purification tasks. This versatility makes agarose beads a powerful toolkit for scientists. It's like having a perfectly adjustable filter for every job. Agarose beads shine in chromatography, a technique where a column is packed with beads and a protein mixture flows through. The beads' pores act as a maze. Large proteins bypass the beads, while small ones get trapped inside, separating them by size. This is called size exclusion chromatography. But agarose beads can do more. Scientists attach special molecules, ligands, to the beads, turning them into custom hooks for specific proteins. Only the target protein sticks, the rest wash away. This process, called functionalization, allows for incredible specificity. Beads can be modified to separate proteins by charge or by how greasy they are. The right hook means only the desired protein is captured. Agarose beads are like custom fishing lures for proteins. This flexibility makes them essential for modern purification. It's targeted, efficient, and powerful. Agarose beads are the ultimate multi-tool for protein purification, adapting to many separation techniques. First, size exclusion chromatography separates proteins by size. Big ones flow past, small ones take the scenic route. Next, ion exchange chromatography uses charged beads to attract proteins with the opposite charge, separating them by their electrical properties. Hydrophobic interaction chromatography uses beads coated with greasy molecules to catch proteins with oily patches, separating them by how much they dislike water. The most precise method is affinity chromatography. Beads are coated with a ligand that binds only the target protein, like a lock and key. Only the desired protein is captured. Everything else washes away. Affinity chromatography can purify a protein in a single step. These methods make agarose beads the gold standard in labs and industry. Their adaptability means scientists can tackle any purification challenge. From simple to highly specific, agarose beads do it all. They're the backbone of protein purification. Without them, modern biotech would grind to a halt. That's the power of agarose. Traditional columns work, but they're slow and cumbersome. Enter magnetic agarose beads, the next leap in purification. Scientists embed iron nanoparticles in agarose beads, making them magnetic but still functional. Instead of columns, you add beads directly to your protein mixture and stir. The beads bind to the target protein, then a magnet pulls them out. No centrifuge or filter needed. Wash, elute, and you have pure protein in under an hour. This batch process is faster and easier than ever before. Magnetic agarose beads are revolutionizing purification. Simple, quick, and incredibly effective. Why does this matter? Because pure proteins are the foundation of modern biology and medicine. Scientists need them to study diseases and develop treatments. Many medicines, like insulin and antibodies, are proteins that must be purified to perfection. Agarose-based purification is essential for making these drugs safe and affordable. Magnetic beads speed up diagnostics and drug discovery, allowing thousands of samples to be processed quickly. From research to medicine, agarose beads are silent heroes. What began as seaweed gel now drives breakthroughs in health and science. Next time you hear about a medical breakthrough, remember the tiny beads that made it possible.